Okay, so good afternoon, everyone. I hope my screen is visible to you.
Yes, sir.
Yes, sir.
Well, let me. Okay, ha. So, from today, we are going to start out with some new set of databases. Okay, and since you are in continuity with the databases that you have explored on NCBI, so we would continue first with the databases that are on NCBI itself. And after that, I thought it would be logical for us to explore the databases that are outside NCBI. Okay, so we are going to talk about these nucleotide sequence databases in this and next few lectures. And these nucleotide sequence database, the major focus would be on reference sequence database, uh, which would be our major point of discussion for today's lecture and maybe next practical. And after finishing up with this, we would also deal with this DBSTS, that is database of STS sequences, and database of EST sequences. So what are STS, what are ESTs, and their implication? We are going to talk in more details of those things in a few upcoming lectures. Okay, so today's lecture will be devoted for discussion on reference sequence database. Okay.
Hello, sir. Uh, hello,
Yes.
sir. Where we need should
Hello.
sir?
Huh?
Am I audible, sir?
Now you are audible. Huh.
Okay, sir. Where we should submit our assignments?
Yes, we will talk about it a bit later. Okay.
Okay, sir.
So, well, let us first discuss about our databases, and then we would discuss about submission of your assignments and all those things, right?
Okay, sir.
So, as I have already mentioned you, today we are going to discuss in more details of this reference sequence database. All right. So first, let us discuss key what exactly is this reference sequence database. Okay, so of course this reference sequence database, what in short we would subsequently refer as RefSeq database. It is you can say it is an NCBI's collection of freely accessible database of naturally occurring sequences. Okay, so this database is responsible to host sequences that are taxonomically diverse, sequences that are non-redundant, sequences that are richly annotated, and sequences that are from different types, like it hosts the sequences of DNA, RNA, as well as protein. You people might be aware that you have got GenBank that deals with DNA sequences, and of course NCBI has got its own different protein database which hosts protein sequences. But this reference sequence database is one of the, you can say, database or resource that has all of the three major macromolecular sequences collected together at one location, that is DNA, RNA, and protein, right? So we would also talk about if there are separate databases of for this nucleotide and protein, then what is the need of having this reference sequence database? That might be something that must be bothering you for time being, right? But just hold on for a few slides, you would get the answer to that question. Okay, so this reference sequence database also includes sequences from plasmids, other organelles like mitochondria or plastid. It contains sequences from viruses, archaea, bacteria, and other eukaryotes that are known. So all in all, it tries to encompass all the type of nucleic acid as well as protein sequences that can, encompass different type of organelles, different type of organisms as such. Okay, so this reference sequence database can be considered, as one of the one stop shopping, you can consider it as, right, कि एक ही जगह पर आपको सब कुछ मिल जाना चाहिए. So let us see कि what exactly is the aims, or what exactly are the objectives of creating this database. Okay, so the main objective of this reference sequence database is for integrating diverse data type into one consistent framework with a uniform set of convention and standards that can help, to unify the information that is, present in different databases. Okay, so everything you would get into a single location, which would have diverse data type. Well, say for example, you can have sequences of proteins, you can have sequences of DNA, RNA, different type of organelle sequences, different type of organism sequences, and everything is to be present in a consistent framework. Okay, so these data types, which might include like sequence information, the genetic information associated with it, its expression information, as well as its functional information. So the aim of reference sequence database is to integrate everything, right? So this initiative supports activities like genome annotation, it supports activities like gene characterization, it also helps in comparative genomics, you can also report sequence variation, as well as it also hosts data on expression studies. Okay, so these were the basic aims which were kept into consideration while creating this resource. Okay, so now let us talk about what is the source of data from which this reference sequence database is built. Okay, so the major source of data used for populating this reference sequence database comes from INSDC. Okay, so INSDC, you might now be quite familiar with this term, stands for International Nucleotide Sequence Database Collection, database collaboration. Okay, so this INSDC, you can say that it is a collaboration between NCBS GenBank, the European Nucleotide Archive, what we call it as ENA, and the DNA Data Bank of Japan, or what we call it as DDBJ. So now the objective of creating this reference sequence database was to provide sequences for each species that are first non-redundant, second these sequences should be extensively cross-linked to different resources, third it must be richly annotated. Okay, so that was the ultimate objective for each species, whatever sequences there are. Okay, there should be a database which should contain some non-redundant sequences for each species that should be extensively cross-linked with other databases, and it should be highly, or you can say richly, annotated. Okay, so one can query this database based on genomic location. That is, if you somehow know, okay, my sequence of interest is located on 6th chromosome of human, then it is also possible that you can search into this database using such genomic location. Or you can also perform sequence-based searching using tools like BLAST, or you can also search it using text annotation. Well, say for example, if you know something about that sequences, like the sequence that I am interested for a particular protein is say 1000 nucleotide long, or say some 3000 bases are there in that nucleotide sequence, then you can, you know, perform searches in such way. Okay, we are going to deal with querying and searching this database in more details in our demonstration and practical session. Okay, but for time being, it would be enough for you if you remember that this database can be searched either if you know the genomic location of the sequences, or if you know the sequence you are looking for. That is, you can perform say sequence similarity searches, and of course you can search it using text annotations. Okay, so now since we say the data to populate this database comes from INSDC, then what is the difference between the entries from INSDC and the entries that are present in reference sequence database? Well, let us make this comparison first, right? So we would compare the INSDC entry with the reference sequence database entry on the basis of multiple attributes, right? So say for example, in case of the reference sequence entry, the accession number includes the underscores, right? So accession number, if an accession number contains an underscore, that would mean that entry belongs to the reference sequence database, while such underscores are not present in accession entries of accessions of the entries that belong to INSDC databases like GenBank or DDBJ. Right. So right from the accession number, you can identify whether this entry belongs to uh, INSDC member, or whether it belongs to reference sequence database. Okay. Now, of course, the databases like GenBank, ENA, or DDBJ, well, these are members of INSDC, while your reference sequence database is an independent database and it is not the member of INSDC. This is most important point that you should know, right? People usually get confused. Okay, so reference sequence database is not member of INSDC, while databases like GenBank, ENA, and DDBJ, well, these are members of INSDC. Okay, so if you now consider the source of sequence, in case of INSDC, well, say for example, in case of GenBank, the source of sequence is the submitter who submits the sequence over there, while in case of reference sequence database, the source of sequence is INSDC entries. Okay. Now, if you consider what is the source of annotation, so the entries from INSDC, well, so for example, whatever is present in a GenBank entry, the annotation are usually provided by submitter, while in order to incorporate annotation, reference sequence database relies on multiple sources. Okay, so the thing is that the data that is present in reference sequence database is inferred from multiple sources, while the annotations that are associated with any sequences in INSDC entry are the ones which are provided by the submitter. Okay. Now, the owner of the sequence record in case of INSDC entry is the submitter, while the owner of the record in case of reference sequence database is NCBI. Yes, anyone wanted to ask anything? Okay, I suppose I can proceed further. So now there is difference in case of owners. Okay, in case of INSDC, the owner of the entry is the submitter, while in case of reference sequence database, the owner of the entry is NCBI. So if you consider the factor of redundancy, The redundancy in INS-DCs is very high. INS-DC databases में redundancy बहुत high होती है, while the redundancy is very low in case of reference sequence database. Why is that? We would come to that in a little while. Okay, so it is possible to archive the experimental data in INS-DC entry, but it is not possible to archive the experimental data in reference sequence database. Okay. One more point to remember is that the data that is or the annotation that are present in INS-DC are not reviewed. I mean, the review of experimental data is not done in case of INS-DC, but that is done in case of reference sequence database. NCBI do not curate the data that is present in INS-DC, but in case of reference sequence database, it is the job of NCBI to curate that data from time to time. Okay, so a regular update schedule is not possible in case of INS-DC entries, but it is possible in case of reference sequence database. So well, now you can get the depth key. There are a lot of factors based on which you can now distinguish key, okay, these are the things that are offered in reference sequence databases while those are not offered in INS-DC entry. Okay, so this is how it is. Okay, now let us talk about the growth statistics and updates of our of our reference sequence database. Well, now the graph that you see over here is the one taken from 9th July 2018. Okay, so now you might have attended the lecture of GenBank, in which you might have seen a graph which shows exponential growth of these sequences that are deposited at GenBank. But in this case, as you can see, the growth do not appear to be exponential. It appears to be linear, है ना? तो यहाँ पर if you remember the GenBank statistics details, the growth of the sequences in database were quite exponential, something like this, right? But on the other hand, in case of this reference sequence database, the growth is quite linear. It is not exponential, right? And for the sake of comparison, I would compare the growth or you can say the number of sequences that were present in 2018 and the sequences that are present in 2021, that is 8th of January, I mean 2 days ago. So as you can see, there is no any exponential growth over here. So there is a reason why there is exponential growth in case of INS-DC entries and the linear growth that happens in reference sequence database. Okay, we would come to that point in a little while. So this database, reference sequence database, is usually updated on daily basis. जो जैसे annotation information available होता है वो वैसे वहाँ पर update हो जाता है, right? So well, now let us talk about why is that there is exponential growth in case of sequences in case of your INS-DC entry जैसे कि GenBank, and why the growth over here is linear. So the main reason behind it is it can be attributed to the fact that whoever wants can submit the data at GenBank, right? But in order to create an entry into reference sequence database, a lot of manual curation is needed to be done. Okay, So due to this, what happens is that the data that is present on the reference sequence database, you can say it is more reliable. Why? Because it has undergone some manual review. Okay, so we would talk about the pipelines and everything which is used for reviewing or annotating the data that is present in the reference sequence database, but this can be considered as one of the attribute. इसमें ग्रोथ लीनियर क्यों है? क्योंकि इसमें मैन्युअली क्यूरेटेड डेटा रहता है और मैन्युअल क्यूरेशन को टाइम लगता है. On the other hand, there is no any review of data that is submitted in INSDC entry. Therefore, whoever wish can submit the data, and therefore the growth over there is exponential. Okay. So now, well, let us talk about the flat file record of this reference sequence database. Okay. So in the next few slides, I will show you a typical entry of the reference sequence flat file record, and you will you would infer that it is quite very similar to GenBank flat file format entry. Okay, so yes, of course it is quite similar to that of GenBank flat file entry, but I will kind of focus on what are the distinguishing features that are present in the reference sequence flat file record that are absent from your GenBank entry. Okay, so that you can distinguish between the two flat file records. Okay, so the first and foremost distinguishing feature of a reference sequence flat file record is its accession format. Okay, you have got a very distinct accession number format that begins with 2 characters. Okay, and it is usually followed by an underscore. Okay, well, so for example, the table that you see over here, okay, so whatever accession number are given, those are usually beginning with, say, some accession prefix which are listed over here, as you can see. Okay, and after each 2 character accession accession prefix, there is an underscore, and after this underscore some definite number or some number or say some character or number might be present. Okay, we would talk about that in a little bit of while, but for time being, you can easily identify based on the accession prefix itself whether the flat file record belong to what kind of molecule type. So say for example, there are at least 7 accession prefixes that belongs to genomic type. There are at least 4 accession prefixes that belongs to RNA molecule type, and there are about 5 accession prefix type that correspond to proteins. Okay, now based on what are these accession prefixes, you can categorize what type of genomic sequence it is, what type of RNA sequence it is, what type of protein sequence it is. Okay, so it is all up to you to explore this table. Okay, and well, say for example, if if the accession prefix is like NP underscore, that means the flat file with accession number NP underscore belong to a protein. Okay, and it can also be associated with either NM or NC accession number. Okay, so in this way you can categorize the molecule type on the basis of accession prefixes as well. Okay, that is what I want to convey you from this slide. Good. Then the another distinguishing feature of your reference sequence flat file record lies in the comment section. Okay, so this comment section from reference sequence flat file record focuses on what is the status of that record. Okay, so each and every record will have some status associated with it so far it is present in reference sequence database. Okay, so the status of the record can be either model. The status of the flat file record can be either inferred. The status can be predicted, provisional, reviewed, validated, or whole genome sequence. Okay, so well now what is the details of annotation that is present or that is offered in that flat file record? Based on that, these status are provided, and you know this status would change as the annotation, or you can say the level of annotation is increased. Initially, the status of a flat file record can be model since it is not properly reviewed. Okay, but this status can be changed to reviewed or validated depending on what kind of annotations are now being added to that entry. Okay, so I would advise you to read what are the different descriptions of the status that are usually associated with this record status types as such. Okay, so what do you mean when we say that the reference sequence flat file record has a model status? What do it mean when the reference sequence flat file record has a reviewed status? Okay, when we say that the record attains a reviewed status, when the reference sequence record, we say that it has obtained the reviewed status when it is, you know, reviewed by NCBI staff or by a collaborator. Okay, so the NCBI review process might include accessing available sequence data and the literature associated with it. Okay, some reference sequence record may incorporate expanded sequence and annotation information. Okay, on the other hand, the model status is given to a reference sequence record when it is, you know, extracted from the genome annotation pipeline. We are going to talk about these genome annotation pipelines and different pipelines that are, you know, implemented for extracting information and providing to that reference sequence flat file record in a little bit of while now, but things would get more clear when at the end of this lecture. But for time being, आपको क्या ध्यान में रखना है कि हर एक रिकॉर्ड को कुछ स्टेटस रहने वाला है, एंड द स्टेटस would be dependent on what is the level of annotation that is present for that record. Okay, validated is the highest status. That means it has undergone initial review, and you know nothing much would change after after it achieves the validated status. Okay, while model status usually changes to inferred or predicted or provisional or reviewed depending on what kind of annotations are added to that subject entry. Okay, good. The next thing that you should know about this reference sequence distinguishing features is its nomenclature. Okay, so this reference sequence database uses the official nomenclature for gene features. Well, so for example, like species or gene name when it is available. Well, if the official nomenclature is not available, it uses the names from INS disease entry like GenBank, or it uses the data from publication. Okay, protein names that are originating from either UniProt KB or enzyme commissions or NCBI staff curation are usually assigned. Okay, fourth and final most important important feature is DBX rate, that correspond to a qualifier for database external reference link. Okay, so this section provides link to other resources of information like whether this protein or nucleotide entry is associated with OMIM, online Mendelian inheritance in man, or whether it is associated with gene, whether it is associated with uniprot, okay, or whether it is associated with the consensus coding sequence CCDS or conserved domain database CDD. So there are, you know, external reference link. So this link would take you to that particular database जिसके बारे में detailed information वहाँ पर रहेगा. Okay. So now let us talk about this simple flat file of your reference sequence database. Okay. So at the very first glance, you might see okay, things are quite very familiar, and you know, these are things that are you know, similar to that of GenBank flat file format. Well, so for example, you can see over here there is a line identifying that this is a reference sequence record. किससे पता चला अपने को? Using its accession number. Okay, you have got 2 characters, then an underscore, and then some number. So this clearly indicate that this is a reference sequence record. Okay, then you have got distinct accession number prefix. Okay, and you can also have some data related to publication, right? In what paper that this particular entry was published. We'll say, for example, it was published in this journal BBA Research Communication back in 2005. Okay, there is also link for PubMed over here. Okay, there is a link for this organism Homo sapien. You would be carried on, if you click on this Homo sapien over here, you would be carried on to that taxonomy database, which is differentially present on NCBI server. Okay, so on and so forth. Okay, so as I have already mentioned you, there is a big comment section in the flat file. एंड दिस कमेंट सेक्शन में सबसे पहले क्या होता है, उसका स्टेटस होता है. इन दिस स्टेटस दिस पर्टिकुलर एंट्री हैज रिव्यूड स्टेटस. Okay, then what is the data source, reference sequence gene information, and sequence history, how many time it has already changed, right? और उनके accession numbers भी यहाँ पर है. Okay, then you have got other details that are present in your GenBank, like उसका assembly details है. You have got indication of partial publication list if there is any. Okay, and this is what I was talking about. These are called as links to external databases. Okay, so if you click on this link, you would be directly carried on to this gene database. If you click on these exons and all these links, you would be carried out or you would be forwarded to, you know, the respective source. Well, so for example, this is the link for DBSNP. Okay, so if you click over here, that particular entry is associated with that single nucleotide polymorphism, or what we call as SNPs. Okay, so whatever data might be associated, it is usually linked with some external resources. This is one of the, you know, biggest strength of using reference sequence database. ये आपके NCBI के GenBank में नहीं होगा. Okay, now let us talk about how data is assembled and maintained at a reference sequence database. Okay, so first and foremost, the data is assembled by extracting the information from INSDC submissions. Okay, so whatever curation, annotation, and computation that would happen on the reference sequence is usually combined with extensive collaboration with authoritative groups that are present in NCBI, or that can, well, they can also, you know, import those annotation from external resources as well. Okay, so we call such people as collaborators. Okay, so the job of those collaborator is to provide information on nomenclature, feature annotation, as well as links to external organism specific resources if there are any. Okay, So if there is no collaboration that has been established, then this job is usually done by NCBI staff itself. Okay. So the comment section of the record would indicate what is the level of curation that is received from the collaborator. Okay. So you can, I would give you an assignment in which you would explore all these things. Okay. So this particular figure is very important. Okay. And it represent on how data, annotations and data are processed at this reference sequence database. So as you can see over here, there are three different pipelines that are usually implemented for curating or annotating the entries that are present in your reference sequence database. Okay, so now the objective of using these pipelines is different for different purposes. Like, so for example, you have got this curation pipeline, which is usually implemented to obtain information, curate and annotate the flat files that are vertebrate in origin, while your GenBank extraction pipeline is usually used for managing the prokaryotic sequences, while annotation pipeline might be implemented for transcript and protein models. Okay, so the very first and very simplest simplest pipeline to understand would be this GenBank extraction pipeline. Okay, तो इसमें होगा क्या कि whenever a new entry is established in your GenBank, what happens is that its duplicate that record's duplicate copy is created. Okay, and then it is tested for validation and a reference sequence formatting. Then it is tested कि whether if it belongs to the prokaryote. Okay, और अगर ये prokaryote को belong करती है, then it would undergo the clustering. People would go on adding names, gene IDs, and after all these processes are done, usually the entry would get the status of either provisional, predicted, or curated reference sequence entry. On the other hand, if it is a transcript entry, right, what would happen is that first the process of there would be alignment of reference sequence and GenBank transcript to genome assembly. Well, now you might be wondering what this genome assembly is, or how do we align to genome assembly and everything. Things would get quite very clear when you study in more details of another subject called as genomics. Okay, but for time being, just only consider that the sequence would only enter in annotation pipeline if it is of transcript type. Okay, that is if it is from it, if it is of RNA origin, right? So if it is an RNA, then there might be certain softwares that would predict the transcript, and by predicting the transcript, and subsequently you can create protein models based on based on those six open reading frames, right? One from one strand, one from its opposite strand, and you can also then select what of these six reading frames, whatever protein sequence that you get, one would somehow match with some entry in protein database, and that particular protein model would be used for further annotation. So in such a case, usually the status would be given as model status in reference sequence database entry. Okay, on the other hand, if the sequence belong to vertebrate, there is a different pipeline that is needed to be followed, and by the end of this pipeline gets in function, initially you can have either provisional, predicted, or inferred reference sequence database status. After curation, these entries can be changed into curated. Once they are curated, they would be having status of either validated, or they would be having the status of either reviewed. So, depending on what kind of pipeline these sequences has come from, you can have different status. Okay, so how do we now access the data from this reference sequence database, and how do you retrieve the data? तो इसके लिए, since it is part of NCBI, of course you have got Antre query system, so you can get access to this reference sequence database using Antre. Okay, in my previous lecture I have already discussed what Antre is. Okay, if you have sequence to match with, you can use BLAST, and there is also a file transfer protocol site which is present over there. If you want to download the reference sequence, if you want to download in bulk amount, you can use this FTP link for accessing the molecular sequences as such. Okay. So here is how it is. You can perform these queries. मैं आपको बताऊंगा कि ये queries कैसे करनी है when we deal with our demonstration section, right? So how do we retrieve using BLAST the sequences from reference sequence database? BLAST हो गया है क्या तुम लोगों का?
No, sir. No, sir.
No, sir.
Okay. Maybe these things would again make more sense when you deal with BLAST in more details of that. But whenever the BLAST searches are performed, there is an option of selecting a database. Okay, so as you can see over here, if you paste a sequence over here, and then you have got option to select the database in which you want to search for sequence similarity, right? So if you there is a separate option for reference sequence, that is you can select over here, okay, I want to blast it into reference sequence database as such. Therefore you can perform either protein as well as transcript or nucleotide sequence blast as such, right? So that would it is an option available over there. You can use sequence similarity search to search in reference sequence database. Finally, you have got FTP site, like you have got BLAST FTP site, you have got genome FTP site, reference sequence FTP site. So this reference sequence FTP site would contain full reference sequence collection, okay, so that you can download all of the sequence and its annotation if you are dealing with very big length project, or you can say sequence analysis stuff. Okay, so these are the important link that I think you should follow. You have got NCBI handbook में एक 18वां chapter है, वो पढ़ो. There is a lot more explanation given over there. Okay, you can also check out the reference sequence databases frequently asked question. Okay, and you can also consider this fact sheet. BLAST तो NCBI आपको मैडम बता ही देंगी when you would learn in more details of this. Okay, so this is all about your reference sequence database. In our next lecture, I would show you a few demonstration and let you know how you are supposed to do your practicals or your assignments. Okay, so this is all for your theoretical session. All right.